So welcome everyone. We don't have that many people today, but we can still listen to Angelee's talk. I think it will be super interesting. I just want to say a few words about Angelee before we start. Angelee is originally from Mauritius, just like me. So I'm happy to be the host today for that seminar. So she has a co-badged MSc and PhD degree in applied marine sciences and oceanography, both from the University of Montpellier and the University of Cape Town, and also Institut de Recherche pour le Développement, IRD, which is in France. And at the moment, she is a postdoc at the University of Bretagne Occidentale in Brest, France, a co-fund Bienvenue and Marie Curie postdoc. She has her postdoc until 2024, if I'm not mistaken. And after that, if anyone knows of anything happening or any job or any postdoc, get in touch with her. And I'm sure she will be happy to hear about anything at the moment. And today, so welcome to her talk today. The title of her talk is The Importance of Using Multidisciplinary Approaches in Studying Micronekton Communities to Inform Future Sustainable Fishing Practices in the Context of Increasing Anthropogenic Pressures. So I see the title have changed a little bit, which is super nice. We're looking forward to hearing your talk, Angelee. Yeah, the floor is yours.
Thanks. Thank you for the introduction. I'm really happy to be here as well to present my work. So like Pavanee mentioned, I'm also from Mauritius. My name is Angelee, and I won't go through, you already did a very good introduction. I wish to thank the organizers for the opportunity to present this work today during this session. And today I'm going to talk about micronekton communities, more specifically, why is it important to use multidisciplinary approaches to study micronekton? So before I talk about micronekton, rather I'd like to say that I've studied micronekton for a long while now, from my master's degree up to my PhD and my postdocs. So what are micronekton? These are organisms of 2 to 20 centimeters in size. They consist of crustaceans, cephalopods, and mesopelagic fishes. And compared to macrozooplankton or compared to plankton in general, micronekton are able to swim independently of currents. And there's actually an estimated biomass of several billion tons in the global ocean. So these organisms, the mesopelagic ecosystem range from 200 to about 1,000 meters, and this is where micronekton lives and dwells, basically. Although some species are known to also live below 1,000 meters. So micronekton are very important in a marine ecosystem because they have several functional roles. They are a key trophic link between top predators such as tuna, swordfish, and lower trophic level zooplankton. So they provide a key link for the transfer of energy across marine food webs. They're also important in the biological carbon pump because micronekton organisms, or at least some species, are known to migrate from sometimes from below 1,000 meters, sometimes from below 400 meters up to the sea surface at night. And at dawn, they'll migrate back to the deeper ocean. And in doing so, they'll be transporting carbon from the sea surface to deeper layers of the ocean. Hence, micronekton also contribute to the export of atmospheric carbon from surface layers to the deeper ocean, hence playing an important role in the carbon cycle in the biological pump, especially in the context of now where there's increasing sea temperature, increasing carbon dioxide in the atmosphere. So micronekton more than ever are playing a key role in marine ecosystems. Now there are increasing interest to exploit micronekton for nutraceuticals, for minerals, as dietary supplements, and also for use as fish meal in aquaculture farms. So several countries have expressed interest. So far, there's not a major micronekton fishery, although krill is extensively sampled is extensively fished for in the Southern Ocean. Countries are still trying to explore different sampling gears and the feasibility of a micronekton fishery. So on the next few slides, I'll go through some of the tools I use to study micronekton and the results obtained. So during my master's project, I used a combination of acoustics, trawls, and stable isotopes to study the migration, traffic position, and the composition of micronekton in two contrasting provinces of the Indian Ocean. So as you can see in this sea surface chlorophyll map, the ISSG, the Indian South Subtropical Gyre, is very oligotrophic. And compared to the East African coastal province, which is more productive due to the action of mesoscale eddies, driving productivity from the African landmass into the middle of the channel. And this is also a region where there are a lot of mesoscale dynamics contributing to the overall productivity in this region. So this has an impact on micronekton communities. As you can see in the oligotrophic region, we have less micronekton. So around Mauritius in the gyre over here, less micronekton compared to the East African coastal province. D and N refer to day and night. So like I mentioned, micronekton migrate every day or every 2 days, depending on the species. So generally you have more micronekton in the water column at night because they're migrating from depths compared to daytime. But as you can see during both day and night, so more productivity, more micronekton in the province closer to Africa compared to the oligotrophic gyre. So I also studied the trophic position of micronekton. So in green, you can see phytoplankton, and then we have zooplankton. Micronekton are over here, and swordfish is at the top. But there's a difference in the trophic, or rather the trophic space. The trophic food web is different in the ISSG and EAFR. As you can see in the ISSG, there's a trophic gap between swordfish and micronekton compared to the EAFR, where we don't really see that gap over here. And that's because in the more productive province, swordfish are usually smaller in size, and they would forage on a wide range of micronekton because these are abundant and they are more readily available. So they would forage on opportunistically on micronekton, anything that they can find basically in the water column. Compared to the oligotrophic province, swordfish tend to be bigger in size. And since it's hard for them to find micronekton prey in this region, they won't target micronekton as much in the ISSG compared to the EFR. So swordfish would preferentially feed on larger size cephalopods, and these were not collected in our trawls, so we missed this gap over here. So this points to the productivity, which is different in the Southwest Indian Ocean, can lead to different abundance and biomass of micronekton prey, and in turn, this leads to different foraging strategies employed by predators such as swordfish. In a region where it's more oligotrophic, swordfish won't spend their energy to look for smaller sized micronekton, which are not abundant anyway. So they would rather focus on these large cephalopods, which would give them more energy, compared to the more productive province, where swordfish would basically feed on micronekton that they would find readily available in the water column. So next, during my PhD, I investigated the patterns of micronekton communities in relation to seamounts of the Indian Ocean. So I studied two seamounts. I studied the Lapuru seamount, which is found to the northwest of Réunion Island. And I studied Madrid seamount, which is found to the south of Madagascar. So again, I use a combination of tools to understand the vertical distribution of micronekton. So I use acoustic data. I use satellite hydrological data to study the environmental influence of micronekton, and I use trawl data to look at the assemblages of the communities. And I also looked at the food web structure using stable isotopes. So again, it's very interesting because we started off studying these seamounts thinking that just like what's been told in literature, that seamounts are regions of enhanced biomass, biodiversity and all that. The results were not what we expected, because as you can see over here, so this shows the acoustic density, so the biomass of micronekton, you can say, during the day and night. So in the oligotrophic region, more productive region at Madrid Seamount, La Perouse Seamount, and I also investigated the micronekton density on the Madagascar continental shelf. And as you can see, again, the more productive EFR province, where there's a greater mesoscale dynamics, these had greater micronekton densities compared to the seamount, to Madrid Seamount. So this points out to the fact that mesoscale eddies of the Mozambique Channel, they drive productivity in this region, and this leads to greater micronekton densities over here. And the effect of mesoscale eddies is rather more enhanced than any type of effect caused by Madrid Seamount itself. In fact, we didn't really see any physical processes at Madrid Seamount which would enhance the productivity and enhance micronekton densities. And it was also due to the fact probably that during our cruise, there was an anticyclonic eddy at Madrid, so which might have contributed to a reduction in productivity and hence less micronekton at the seamount compared to the more oligotrophic, compared to the more productive province over here. And but then we did see a difference between Madrid and La Perouse. La Perouse is located in the oligotrophic ISFG province, and the micronekton density is less at La Perouse compared to Madrid, which is still located outside of the more productive Mozambique Channel, but then close enough to Madagascar and close enough to the site of eddy generation and eddy transport. So more micronekton over here compared to the oligotrophic La Perouse. So basically, this is what we found. Cyclonic eddies are. I also investigated the effect of a cyclonic eddy and an anticyclonic eddy on micronekton. So this shows the cruise transect. Actually, Femi took part in that cruise. I think it was back in 2016. So during this study, these points here reflect CTD stations, but basically I analyzed acoustic data along the west-east transect and south-north transect, spanning an anticyclonic, cyclonic eddy. The seamount flank is over here, and the seamount summit is right here. As we can see, cyclonic eddies in overall are more attractive for micronekton compared to the seamount itself. So these are just the micronekton densities in the whole water column in white in the deep layers. The deep layer is below 400 meters. The surface layer is the sea surface from the surface to 200 meters. So here again, we can see that the seamount, Madrid Seamount, does not have an enhanced micronekton acoustic density compared to the cyclonic eddy. And we've seen this before. La Perouse Seamount in the ISFG province has a lower micronekton densities compared to Madrid. And that's because Madrid is located in a more productive region compared to La Perouse, which is located in oligotrophic gyre. So we also observe the diavertical migration of micronekton in our data. So as you can see here, so black refers to nighttime, and then we're transitioning to dawn, and in gold, this is daytime. So as you can see, micronekton is mostly located at the sea surface at night. And then during at dawn, they would migrate to below 400 meters. And we've seen that micronekton do not all migrate at the same time. You have different communities migrating, well, delaying the migration kind of in the sense that they would migrate sequentially from the surface to the deep ocean. And we saw that there were communities which were associated with seamount summit and flanks. So although these seamounts did not show enhanced biomass or enhanced micronekton densities as we would have expected, well, we saw that they contain specific communities of micronekton that you wouldn't find anywhere else. For example, this species over here, Diaphus subobitalis. Well, Diaphus is a genus which is pretty common across the whole globe. this particular species, Diathos suborbitalis, has only been collected at seamounts of the Indian Ocean. So we collected it at La Perouse and Madrid seamounts along the flanks and the summit each time. And also they were mostly collected at the equator seamount in the Indian Ocean. So this points out to seamounts being attractive sites for specific species of fish. So the Diathos suborbitalis, also we caught an epibenthic fish, the Cocheolus japanicus. So this only mostly shows that, as you can see, the Diathos was mostly collected at the flank of the seamount over here. And we also found that fish which were found at the seamounts, be it La Perouse or Madrid, they would have pretty much the same trophic level of 4, irrespective of their size, which is pretty uncommon because usually you find an increasing relationship with size. So as with increasing size, fish would have a greater trophic level. But here, whether they are smaller size fish or larger size fish, they would all have a trophic level of 4 at these seamount ecosystems. So this leads us to think that some specific fish species would associate with seamounts due to the enhanced availability of prey, or due to the crescent shelters offered by the topography. Or in the open ocean, maybe they have to swim constantly, and then maybe they are less. They're more, what's the word? They're more prone to being predated. Whereas at seamounts, they can pretty much find a wide range of habitats where they can hide from predators, while at the same time benefiting from currents bringing food over the seamount for them. So that's maybe why these species would preferentially associate with seamounts. And the fact that they all have the same trophic level at seamounts, irrespective of their size, points to the fact that they pretty much all may be feeding on the same type of food. So seamounts being topographic elevations from the ocean floor, they might, well, usually they impact the currents around the seamounts a little bit like mountains, which had an impact on wind. The same thing happens in the ocean, except that seamounts, well, as a topographic elevation, have an impact on ocean currents, and the ocean currents will bring food to the seamount ecosystem, and the micronekton living and dwelling there mostly would be feeding on the same type of prey at these seamounts. Hence, the micronekton or the species associated with seamounts having the same trophic level, irrespective of their size. So also I investigated the trace metal concentrations in micronekton of the Indian Ocean. So I had 4 sites, La Perouse and Madrid seamounts again. I investigated trace metals around Réunion Island and the Mozambique Channel. So these range of mercury values are pretty much the same as in other oceans. So the Indian Ocean is not that different compared to other regions of the world. I also saw that squids have lower median mercury concentrations compared to crustaceans and fishes. And there are several reasons why that might be the case. One reason being that, unfortunately, we couldn't sample the same tissue from these organisms, since the squids, we mostly sample the mantle, fish, and crustaceans, we sample the white muscle to do these analyses. So that might be one reason why. And other reasons may be that these organisms don't metabolize or don't metabolize or excrete mercury the same way, hence the differing concentrations in mercury in them. The values are not that different as what we find in other oceans. And as for the other trace metal concentrations, we found slight but significant differences between the Indian Ocean sites, so La Perouse, Madrid, Mozambique Channel, and Réunion Island. Overall, we found that mesopelagic organisms in general are rich in iron and zinc, and they have low levels of other toxic metals such as silver, arsenic, mercury, lead, selenium, etc. Selenium is actually not a toxic metal as such. It is used as a detoxification mechanism by combining with mercury and forming a complex So this points to the fact that if ever there's mesopelagic fishery, mesopelagic organisms would be able to provide iron and zinc as minerals. So during my current postdoc, I use a wide range of tools, from satellite to high-resolution moving vessel profilers to SADCP, TSG data, and acoustic data to investigate the different groups of micronekton which are impacted by sub-mesoscale features. So acoustic data in general, this shows an echogram. Which just shows you basically the intensity of the acoustic signals. So you have high signals, so more stuffs in the water column, I'd say, if the color is lighter, and in black is low acoustic signals, so less organisms in the water column. As you can see from this, it is quite hard to understand what are the specific type of organisms which drive this response. We can only say that there's high or low acoustic densities, like I've been saying from the start of this talk. But we can't really say if there's more fish or squids or crustaceans. So during my current postdoc, I've developed and implemented an algorithm to try to tweak out information about the different types or groups of organisms that comprise this acoustic signal. So most of the work involved extracting the signal, doing a clustering of the frequency differences, because in acoustics we use different frequencies, and each frequency may see organisms of different sizes and also different types of organisms. So by working with the frequency differences, we can backtrack and try to find out what groups of organisms comprise this acoustic signal. So here I just did a clustering of the frequency differences, and I projected them in a 3D space. And so during my study, I found 4 groups of organisms. I've just labeled them eco class 1 to eco class 4. And then I investigated the influence of eddies of the Mozambique Channel on these 4 groups, acoustic groups, I call them, or eco-classes. So this is the cruise transect that I used, which spanned an anticyclonic eddy and a cyclonic eddy in the transition zone. So I identified 4 groups of organisms, and these groups possibly represent fluid-like organisms, so ophysids or of small and large sizes, organisms with small gas bubbles, gelatinous organisms, organisms with gas-filled swim bladders, such as fish. And these, during my study, I could see that these specific organisms showed diavortical migrations, and also large fluid-like organisms, such as ophysids. So this study here is still work in progress, and pretty much it's to better try to extract other types of information from the acoustic signal, trying to find groups of organisms and to relate them to the environment. But then significant knowledge gaps still exist. We still don't know the biodiversity of micronekton. New species keep on being identified. There's also we also have trouble with the taxonomy, because a lot of species, well, there's a lot of cryptic species. DNA methodologies, DNA barcoding is shedding new light on that, but there's still a lot of work to do. We know virtually nothing about the life cycle of the different types of micronekton or the different species. We don't know a lot about the trophodynamics, the reproductive patterns. There's still huge knowledge gap regarding the abundance of biomass. Several studies say that there are billions of tons of micronekton, and the values can range from 1 to 16. So it's quite a pretty rough estimate. We don't know for sure yet the exact estimates of micronekton. And we're still discovering new things about the habitat ranges of each species of micronekton. So this is particularly important given the threats that micronekton are starting to face. such as plastic pollution. Some studies have pointed to the fact that micronekton have plastic in their gut contents. And currently, I have a master's student who's looking at the gut content of fish in the Indian Ocean to see if we find plastic, microplastic in their stomach compared to the studies which have been conducted before in the Atlantic and Pacific. So we don't know yet. Maybe, hopefully, we won't find any plastic, but well, I'm not too optimistic. And also, micronekton, we've seen that they can be potentially, modeling studies have shown that they can be impacted by climate change. So the range of micronekton would shift, the latitudinal range, and also their depths in the water column would shift if there's an increase in temperature in the future. They're also impacted by deep sea mining, and also with oil pollution impacts the biomass of micronekton. And fishing incentives. So given all the knowledge gaps about micronekton, it is still quite dangerous to give fishing or to think about fishing the mesopelagics, since there's still so much things that we still don't know about these organisms. And that's because the reason that why we have so many knowledge gaps is because traditional tools that we use to investigate micronekton, are limiting. So most studies are using trawl nets and active acoustics. And trawl nets, they have huge limitations in terms of escape of organisms. They're time consuming, so usually we can only do a limited number of trawls. And they only provide a snapshot of the communities present at any particular time. And a single, especially as well, we get we only can catch organisms present at that point in time and at that location. While active acoustic, they provide continuous data. The problem is that we cannot really resolve the taxonomic composition of the micronekton fauna. So you have to build algorithm, train the algorithm to try to identify the groups of organisms present in the water column. And these lead to a whole lot of other issues, because it's really hard actually to relate the acoustic densities to groups of organisms. So now we're starting to think about using DNA techniques. These are promising but challenging. So the idea is that species, we would sample a tissue, get the DNA sequence, and that's pretty much like a barcode that we can use to relate to any particular type of species. And that's how we found out that there are a lot of cryptic species. So one species of, I don't know, Daphis suborbitalis in the Atlantic, the DNA sequence might not be exactly the same as the ones in the Indian Ocean. So there's still a lot of work to be done in that field. And developing countries face substantial barriers in participating in deep sea research. That's an issue, and that's because deep sea research, well, we usually use ships, vessels, which are not always that accessible to developing countries, and also all these new types of techniques, DNA techniques, eDNA work is really, really expensive, and finding funding for that is really difficult as well and challenging. But it is important to develop and use multidisciplinary approaches to study micronekton, given all the pressures that these organisms are facing and will face in the future. So all these areas are areas which need to be investigated. So the taxonomy, there's huge work to do there. We need to really work on the biodiversity of micronekton globally, especially in the Indian Ocean as well, because we don't have that many information on micronekton. Most studies are mostly conducted in the Atlantic and Pacific. In the Pacific, NOAA and the Woods Hole Oceanographic Institute, they're leading really big research programs on mesopelagic communities and micronekton. But the Indian Ocean, there are really a few groups. Only my group actually studying micronekton in this region, and we have limited funding, so there's still a lot to be done. Before thinking about doing fishing for micronekton, we need to assess the abundance of biomass. having rough estimates between 1 and 16 billion tons is knowing nothing how these organisms, how the stock would regenerate, is a big knowledge gap. So we still don't know a lot about the distribution and migration of micronekton. These can be resolved using acoustics, but again, we need more data and new ways of studying the ocean as well. If we can't access research vessels, we need to think of ways to develop autonomous platforms to study, to get large amount of data sets within reasonable cost. And then investigate, well, work on the life cycle, gut content, otoliths, and all these different techniques. There's still a lot to be done in terms of mesopelagic work in this region. So I guess it's not possible for any single team or any single country to work on that. So there's the need for international collaboration, development of research programs, and also development of training opportunities to enhance capacity building. Most of the projects related to the Indian Ocean, people are either working on physical oceanographic modeling techniques or working on megafauna or phytoplankton, but there's a huge gap in terms of micronekton. People don't seem really to be studying that trophic level that much, and there's still so much that we need to do and to learn. So the current project I'm currently, or my next project is called NEAT, which stands for Micronekton Diversity, Distribution, and Abundance using environmental DNA acoustic controls. So for the first time, I'll be investigating eDNA collected using 5 techniques. So eDNA samples collected in the Indian Ocean to try to shed light on the diversity of micronekton in the Indian Ocean. And I'll couple that with acoustic and troll data. So that's it from me. Thank you very much for your attention.
Great, thank you very much, Angelee. I see that there's a lot of unknowns and uncertainties, and I hope that from these, potentially you could open some new funding opportunities for you to get a more deeper understanding on how things work, especially in the South Indian Ocean. We all know that the Indian Ocean is not as studied as the Atlantic and Pacific Ocean, but hopefully we will get there at some point. Thank you very much again, and I will open now the floor for questions. We have 3 questions. We have a few questions actually in the chat. I will first start by the chat questions, and then we can open the floor to other people to ask questions in person. So we have a question from Himanshu Saxena, which says, what body parts of fishes and nekton are used to measure their isotopic signature?
So for fish, we use the dorsal muscle to look at stable isotope signatures. So, okay, so was it fish and?
Nekton.
Oh, nekton in general. So for cephalopods, we use the mantle, and for crustaceans, the kind of like the abdominal muscles.
Okay, I hope that answers the question. Another question from Saloni Mishra. By using which method or technique the microplastic is found in gut of the fish?
So for this, you extract the gut, and there's actually a protocol where you digest the gut contents using either potassium hydroxide or sodium hydroxide. So now there's a lack of consensus within the community on which solvent to use, but I'm using the same protocol as a colleague of mine in the Atlantic, so we can compare our studies in the future and maybe collaborate together. So I'm using sodium hydroxide, and then once you you've digested the gut and you get the microplastics, so then there's a first step where you separate the contents. So the microplastic, you note down the morphological characteristics, the length, the size, the weight, the color as well. because we're trying to see if whether these fish, whether there's a relationship between the amount of colored plastic, if they would tend to eat more of plastic of a particular color or particular shape or size or not. So we're doing a lot of morphological trait-based analysis. And then, so if we do find a lot of plastic, microplastic, these would be sent to a lab to do FTIR analysis to get the plastic polymer type as well.
Great, thanks, Angelee. We have two more questions in the chat, and before reading those, I would ask if anyone has any questions who would like to ask in person. You can just unmute yourself and ask the question. No. Oh, you do.
Hello, Angelee. I'm Meera. Actually, I had done my PhD in mid-topics. I'm working in Indian Ocean, like Southwestern Indian Ocean, Arabian Seaside. So I was eagerly waiting to meet you. Like I had read your papers, that recent papers and all, and now we have a project like fishery habitat assessment of mesopelagic fishes, mainly concentrated in Arabian Sea. So really, I would like to have a collaboration with you. So this webinar is very much helpful for me, and I had taken your contacts. I hope I would like to personally contact you. Like I have a lot of samples with me, and I really need a help. So this is I would really thank this coordinators for having this webinar series. Thank you.
Yeah, well, I'm really glad as well to hear that you're working on methodologies, Meera. So it would be great, actually, yeah, if you feel free to get in touch anytime. And yeah, well, yeah, we really need people to collaborate and work together on this. There's too much for any single team or person to work on. So yeah, thank you for getting voicing out, and yeah, I really appreciate it, and looking forward to hearing from you and what you're doing.
Okay, thank you very much. Okay, surely I'll contact you.
Oh, feel free.
Great, thanks, Meera. I'm happy that this webinar is providing ways of networking for people from different parts of the world.
Yeah, that's awesome.
Great. So I will get on to the next question from Abul Qasim. How squid samples were preserved and processed for carbon and nitrogen isotope measurement after retrieving from water body?
So once we collected the squids from our tour, they were all identified taxonomically by experts, and then we sampled the mantle. They were stored on board at -20 degrees, and it's back at the lab. We did I freeze-dried them, and then they were ground, and then lipids were removed because they might impact the isotope signatures, and then we did a mass spectrometry. That was rapidly the methodology that we used to get stable isotope signatures from squids, and the same type of methodology was used for fishes and crustaceans as well.
Okay, I hope that answers the question. The next one from Mansi Gupta. Hi, Angelee. Thanks for the interesting talk. Apart from the limitations of active acoustic, what are the challenges you face when studying micronectons given the small size scales of organisms? And how is the uncertainty in bulk value in this technique?
So do you mean bulk value? Do you mean the biomass or the density? I'm just trying to...
Yeah, I meant the biomass. Can you hear me?
Yeah. I guess the limitation actually is to tease out the micronekton signature from other signatures. Acoustics is a technique where basically you send an electromagnetic signal in the water column, and the signal gets reflected by the swim bladder of fish or the gas inclusions within gelatinous organisms, but it can also get reflected by bubbles, gas bubbles. So then during the acoustic data processing, we have loads of steps to process the data to remove any unwanted signal, any background noise. which are not really biological. So these gets removed during the processing. And then also we usually use a threshold, a cutoff point, where we say that we know that from literature that this threshold would, let's say it would discriminate, it would remove signals which are not from micronekton, and it would, so then we'll get, so that's what we do to get the biomass or the density of mesopelagic communities. By using threshold, by doing a treatment of the acoustic signal to remove any type of signal which are non-biological and any type of signal which are not related to these organisms. But there's still a lot of uncertainty in this technique, so that we always use standard deviations when we talk in terms also when we publish about acoustic, we always give a mean, but then with the standard deviation, because you try to remove as much noise as possible and do as much treatment cleaning as possible, but yeah, should be wary. So that's also so that's why acoustic is a big limitation, actually, because we get a signal, but it's really hard to relate it to specific organisms. It's really hard as well we are able to quantify the uncertainty by using so acoustic has progressed a lot throughout the years. So but then, yes, so there's still a lot of uncertainty. So that's why I'm more transitioning now to eDNA as well. I don't think we'll be able to do quantifications with eDNA because there's still a lot of unknowns. But at least I want to get the biodiversity the biodiversity right.
Thank you.
One last question on the chat from Nazia Ahmed. Is there a particular carbon and nitrogen isotopic value for each group of fish or nekton as shown in the plot of delta C versus delta N? And how is the value defined?
So I think it's going back, it's this plot. I didn't show, so this plot is just a biplot. But what we usually do as well is that we plot, so if I plotted box plots of the different groups, sorry, my cat is trying to walk on my keyboard. So there is a particular, or I would say rather a range of values for each group for whether it's fish or nekton or crustaceans. So usually fish have trophic levels of 3 to 4. Crustaceans have trophic levels of around 3. And nekton, it really depends on whether they are large nektonic squids. These have trophic levels of sometimes 4 or 5. And smaller nektonic squids have trophic level of 4, just like fish. And this value is usually defined, the trophic level is defined using calculations. So there are various calculations in literature. Predominantly, there are 3 calculations. So these use the carbon and nitrogen isotopes well, each of these calculations use different, they're quite different, each one of them, but they use carbon and nitrogen values. And so I've tested these calculations and pretty much they give the same they did give the same range of values for fish, crustaceans, and squids. Yeah, I hope this answers the question.
Good, thanks, Angelee. Yeah, I hope that answers the question. If not, please unmute yourself and ask any further questions. I have one question. Maybe that's yeah. So we have from that chat. Okay, thank you. So I guess that answers the question. I have a quick question. If you go 1 slide or 2 slides forward. Here, exactly. Yeah, sorry. Just 1 slide back.
1 slide back?
Yeah, one more. Yeah, here, this one. So, for example, when I'm trying to compare these different regions, especially the Mad region and the La Pérouse seamount, right? So, what strikes me is that for La Pérouse, for example, you have only the surface layer. Is that because it's just very shallow, or how do you define your? I think the surface layer is 0 to 200 meters, right? Something like this.
Yeah. So there are several possible reasons for that. The fact that it's very shallow, and also because Madrid extended the summit was at 240 meters. So then so if you try to, so maybe here. So this is Madrid. It's at the summit is at 240 meters. So let's see, this is a water column. So if I take the 200 meters, and as you can see, Madrid is basically kind of like just like a spike, I'd say, coming out of the sea surface. So if you take the sea surface over here, you'd get the 200 meters around here. La Pérouse is very, very shallow, but then it extends down to 5,000 meters, while Madrid is only to, I don't know, 3,000 meters. And La Pérouse is really extended as well. If you look at the shape and it's not a spike, it's actually a horseshoe shaped seamount. And if you take it's and like I said, it's very shallow, extending very deep in the water column. And if you take the basically you would, if you look at the water column, you'd get mostly the surface, because all the rest, like, it's so extended that you wouldn't really get the deeper layers covered that much. Because try to imagine the seamount coming out, like it's filling that space basically, where the deep is below 400 meters. So that's the seamount itself is filling the space. So you would have micronekton. Typically what they would do at night, they would migrate to the surface from the summits over here. They would migrate distances up to the surface. Some of them would stay at the seamount top, and during the day, usually micronekton would migrate back down. So these are mainly seamount associated species which would do that. They would migrate from the flanks up to the surface and migrate back down along the flanks during the day. Other more oceanic species, they might have varied strategies. They might do that, or they might just get carried with the currents, and micronekton have the potential to swim as well. So unless unlike the species which would preferentially stay at the seamounts, a large number of species, the oceanic species, and they would basically just migrate anywhere in the ocean. But coming back to the question, I think it's probably mainly because of the topography of the seamounts. It's really extended, so that's probably why you have more surface, because the seamount is blocking the deeper layers basically.
Yeah, cool. That makes sense. Thanks, Angelee. Okay, do we have any last question from anyone who's still here who would like just to unmute and ask? Then I will have one last question for you, Angelee. So I see in, I don't know which slide was it, but those transects that you had between eddies and within eddy and between eddies.
Yeah.
I think it was further.
Yes.
Yeah, exactly. So do you see more micronekton within an eddy, acyclonic or anticyclonic eddy, or do you see is there more abundance, or in terms of density, is there much more density on the flank of an eddy?
Yeah, it was actually, we found in this study, I used, so like I mentioned, acoustic data gives you several frequencies. So in this case, we had 5 frequencies. And I looked at the density by frequency and across each zone, and it really depends on the frequency. So like I said, each frequency would be able to see different size class of organisms. So with some frequencies, the lower frequencies, you would find more within the cyclonic eddy, and in some frequencies, you would find more in the anti-cyclonic eddy. And it's really hard actually in this study to look at the effect, whether it's an edge or a center of the eddy, because as you can see, so this is a composite image over several days, but the eddy was moving, and to define an actual boundary is very hard. So I just looked at the effect, anticyclonic, cyclonic, and transition zone. I didn't really look at core of the eddy or edge. But then I can say that depending on the frequencies, on the size class of the organism, so some of them were found in greater densities in the anticyclone and some of them in greater densities in the cyclone. And the migration of the micronekton were different as well. In general, I found a greater proportion of micronekton migrating within the transition zone, which was quite, well, we were not expecting this kind of, but then it does make sense in the sense that maybe this is such a productive site, and also the whole dynamics of the two eddies, maybe it might be helping micronekton just moving up the water column. Maybe that's why you see a difference in the migration strength of micronekton in the transition zone compared to the core of the eddies themselves.
Yeah, that's super interesting because there's more and more, well, now there's more and more studies on sub-mesoscale features, sub-mesoscale dynamics, and there are a few studies which show that on the flank of eddy you have more, you have enhanced vertical velocity. So this could potentially be in line to what you're saying here, with more migration of micronekton on the flank of eddies. So yeah, that's super interesting. And also these like your sea level anomaly are from Aviso, which is not which is, like you said, interpolated and then average over a few days. So it's really hard to really identify where the flank of the eddy is or not.
Yeah, that's why I even categorizing into cyclonic, anticyclonic, and transition zone, there's loads of debate within the community about how you define a front. Where does the cyclonic end and the anticyclonic start? So we had to use, so that's when we use a range of data set, TSG, ADCP, and UVP, to really try to see when there's a shift in currents, when there's a shift in temperature salinity gradients. And that was hard enough. And in my data, actually, I couldn't really distinguish from the TSG, from the MVP. It was hard to distinguish between the core and the flank of these eddies. So it's really subjective in the sense that you see a sharp transition when you move from the cyclonic transition and then to cyclonic. You don't see that sharp transition when you have a core and the edge of the eddy. So that's why I didn't really want to go into. It would have taken a lot more work, I think, and it was not within the focus of my study. It was basically try to identify the acoustic groups and relate them to the environment. But these data are open for collaborative work if people would be interested to dive more into these physical oceanographic data. So, and also during the cruise, we found loads of internal waves. We found loads of really cool sub-mesoscale features. So if anyone is interested in this type of work, I mean, the data sets are there. So yeah, there's so like Femi mentioned at the beginning, there's I'm looking for a job next year, and there's loads of potential for collaborations and between different teams and different people.
Nice, thanks, Angelee. Yeah, anyone who knows of anything happening, yeah, please get in touch with Angelee. Okay, then I think we are almost close to 1 hour session now. Thanks again, Angelee, for giving this really interesting talk, and I would like to thank everyone for attending. And we will see you all again in 1 month for another set of webinar. Great, thanks a lot, Angelee.
Thank you. Thanks.